Thank you. Okay, is it? Yeah. So thank you for the nice introduction and thank you for the chance to present my work here today. So I'm Henriette, I'm from the ChairofPhytophathology. I'm the group of Martin Stegmann, and today my title is Regulation of Plant Hormone Susceptibility by the receptor kinase FERONIA. So let's talk on. It's not maybe. I think it's because you got it. Okay. Why is it? No, it's not. No, it's not. Yeah, I think I just cannot go back, but then I just don't do mistakes. What does this? But you don't have the. Now you have the present. Now it looks better. Oh, try it. Yeah, cool. Okay, now that we solved the technical issues, let's talk about plants a little bit. So when we look outside today, in spring, everything is green, everything is flowering, it looks really nice. And that is despite of the fact that plants are all the time confronted with different stresses. So abiotic stresses like dry, drought, or high UV radiation, but also typical biotic stresses, meaning different pathogens that try to infect the plant. And this can be bacteria, but also fungi, viruses, nematodes, and so on and so forth. And all these pathogens, they can induce severe plant diseases that then, of course, reduce plant growth, can also reduce the yield or the quality of the food product. But the plant is not just sitting there waiting to be infected, but it has evolved also an immune system. But the plant immune system functions a bit different than what we know from humans. So you don't have specific immune cells that we have, like B cells or T cells. But in plants, it is that every plant cell is equipped with components of the immune system. And here, the first layer of plant immunity is called pattern-triggered immunity, or short PTI. And it is, or very important here, are these pattern recognition receptors. And pattern recognition receptors are receptor kinases that sit on the plasma membrane of the cell, and their extracellular domain surveys the apoplastic space surrounding the plant cell. And if now a microbe starts to attack and tries to infect the cell, it produces microbe-associated molecular patterns. So that are conserved molecular structures that derive from the pathogen and that can be recognized by these pattern recognition receptors. And a very common or very well-studied pair that a lot is known about is the bacterial FLAG22. That is a 22 amino acid long peptide that derives from bacterial flagellum, so this little tail here at the end, and it is recognized by FLS2. And upon perception of the elicitor, the pattern recognition receptor forms a complex with a co-receptor. In that case of FLS2, it's FLAG1, and that leads to a very complex intracellular signaling cascade that finally culminates in the defense responses, and that is a first layer of immunity trying to fend off the attacking pathogen. But of course, the plant cell also has more receptor kinases than just pattern recognition receptors. And these receptor kinases, they are very similar in structure to the pattern recognition receptors, but they don't perceive mumps, but endogenous peptides that are produced by the plant to regulate growth, development, or the response to different abiotic stresses. And of course, these two pathways, they don't just exist in parallel in the plant cell, but there is a constant crosstalk or cross-regulation between immunity and plant growth and development. And that also means that by these receptor kinases that perceive per se plant endogenous peptides, they can also modulate whether a plant is either more resistant or susceptibility towards certain pathogens. And one receptor kinase, which I'm focused on in my talk and also in my project, that has exactly this function is FERONIA, and FERONIA perceives Ralph peptides. So let's have a closer look here. FERONIA belongs to the family of Catharanthus museus receptor-like kinase 1-like proteins. And it has its typical structures. You can see the extracellular domain, transmembrane domain, and an intracellular kinase domain. And as I already said, it binds the RALF peptides, which stands for rapid alkalinization factors. And it does so in a complex with the co-receptor LLG proteins. And this RALF FERONIA signaling is very important for the plant. So it really is a major signaling hub. And you can see this because the FERONIA mutant, it has, for example, this very significant dwarf phenotype. So the plants are much smaller. They also produce less root hairs, and also the trichome production is impaired. Further, we see reduced plant reproduction in the FERONIA mutant, and also increased sensitivity of the mutants to the plant hormone ABA. But for all this, I don't really want to explain much more. It's just to give you an overview in how many different physiological functions FERONIA is involved. And that is just a few examples, so there's much more. And of course, I would not have talked about plant immunity so much in the beginning if FERONIA was not also involved here. And that's what you see here. So we have here bacterial infection with the pathogen Pseudomonas syringae. And we see in the mutant, we have higher amounts of the bacteria compared to the wild type for 0 or to a complementation line. And that is because FERONIA has a scaffolding function in pattern-triggered immunity. So it facilitates the complex formation of the pattern recognition receptor FLS2, which with its co-receptor, which then increases the defense responses, which we don't have in the mutant when FERONIA is missing, or we see this increased susceptibility. And now we come to powdery mildew that you already learned a little bit about in the introduction. So that's a fungal pathogen. And what is interesting here now is that we see the opposites from what we see in the bacterial infection. So powdery mildew produces these very typical symptoms here that are this white powder, and you probably have also seen it in nature sometimes, and it just consists of the mycelia and of the conidiforms growing on the plant surface. But we do not see this here in the FERONIA mutant. So that means, in contrast to the bacterial infection, where the mutant is more susceptible, here we have increased resistance in the mutant, which is also depicted here by qPCR, where we see reduced amounts of fungal DNA in the plant samples. But it is not really known so far, unlike for the bacterial infection, where we know it is the scaffolding function, why FERONIA really has this susceptibility function during powdery mildew susceptibility, and what are the underlying mechanisms. And that is where we thought that we could use untargeted co-BioMS approach to find different interactors that might explain which mechanism is underlying here, and maybe we also find interactors that really explain the differences in, on the one hand, inducing resistance, and on the other side, inducing susceptibility. So that is what we see. On the one side, we have resistance, on the other side, susceptibility. And that's what we did. So we had FERONIA GFP plants and LTI6P GFP plants. So that is another protein that we used as a negative control. And we infected these plants with powdery mildew and also with the bacteria, and then had the corresponding treatment controls to each of the infections. And from that, we extracted the protein, and I did a GFP pull down with GFP magnetic beads and trapped magnetic beads. And we sent everything to MSMS analysis, which was kindly done by Tina and her team. And we're very thankful because we had some quite interesting candidates that we found here. So what did we find? We find 44 general interactors. And by general interactors, I mean proteins that are significantly more found in the GFP untreated sample compared to our negative control LTI6B. So they are just found to be interacting with FERONIA. Then when we look at the powdery mildew infection, here we find 7 proteins that are significantly interacting more with FERONIA in the powdery mildew infected samples compared to the untreated control. And now if we look at the bacteria, we find a 307, so a much larger number of proteins that interact in the bacterial infected samples compared to the silver control. But yeah, then I just looked into the candidates here. And I, of course, cannot tell you about all these proteins, what they do and why they might be interesting for us. But I will concentrate on some candidates that we found for the general interactors and for the powdery mildew infection, because that is what my project is mainly concentrating on. And for the general interactors, we nicely found other members of this Catharanthus rheseus receptor-like kinase 1-like protein family that FERONIA belongs to. And it is very interesting for us because here, for example, HERG1 It is also known to be involved in cell elongation during growth. And MEDOS1 here and LED1, they are known to work in a complex together during cell death and autoimmunity. And for me, actually, the most interesting candidate is this one here, because it is so far only an ATG number. There's no function known about it. And I think that's really nice, because I hope that we can maybe give it a function during powdery mildew susceptibility, and by that find out what this protein actually does in the plant. And another group that we found here are plasma membrane localized proton pumps. And that is for us interesting because they really nicely fit in our current working hypothesis. Because what is known is, as I said, these RALF peptides, they induce apoplastic alkalinization. Because when they bind to FERONIA, it happens that you have this phosphorylation here, and that inhibits the proton pump function of these AHA proteins. And during powdery mildew growth on barley leaves, it is known that you also see this spiking in the apoplastic pH. So our current hypothesis is that maybe this RALF FERONIA dependent pH regulation is needed by the fungi, or even used and modulated in order to successfully colonize the plant surface. And we also have some data already that underlies this hypothesis. So I tested a mutant that has a constitutively active AHA1. So that is kind of what we expect also for the FERONIA mutant when we don't have this inhibiting phosphorylation here. And we see, similar to the FERONIA mutant, we have increased resistance depicted by reduced amounts of fungal DNA on the plant. And the same I see when I treat the plants with fusifucin. That is a toxin that has kind of the same effect. So it also leads to constant activation of the proton pump, so acidification of the plant apoplast. And then to our powdery mildew infected samples, here we had two candidates that were really interesting for us and that we want to work further on. And that is on the one hand SAR1, and SAR1 is a NLR protein. And NLR proteins, they are important for the second layer of plant immunity. So I really in detail now explain the first layer, PTI. There's the second layer. I don't want to explain it that much, but it is very interesting for us because it is also involved in immunity, and it's nice for us that it shows up here. And the second is WOT1. You can see it's unfortunately not fully significant, but it's still popped up, and it is very interesting for us because it's a pectin binding receptor. And pectin is a compartment of the plant cell wall. So for basically the first barrier that the fungus has to penetrate through in order to infect the plant. And it is also involved in pathogen response and cell expansion during plant development. And with that, I already want to come to our future perspective. So what do we want to do with that information? First of all, we want to really characterize further the function of these proteins. And that we want to do by generating CRISPR-Cas mutants on the one hand of this protein family, also having higher order mutants, so knocking out multiple of these proteins in the wild type and also in FERONIA to see if they have also a function in susceptibility, or maybe if they also work in contrast to FERONIA and have a function in resistance. And then, of course, we need to confirm the interaction with FERONIA that we've seen now in our BayBioMS data. And I'm generating currently a WOT1/2 double mutant also to see how that is involved in powdery mildew susceptibility. And the second is that we further want to analyze this RALF FERONIA dependent pH modulation and what this what exact role this has during powdery mildew interaction. And for that, we use a apoplastic pH sensor. This is what you see here. So it is GFP tagged to RFP, and with that, you can survey the apoplastic pH in the plant under the confocal microscope. And also I'm doing different infiltration experiments with buffered medium to see how the buffering of the apoplast influences powdery mildew susceptibility. And with that, I'm at the end of my talk. I want to thank Martin for being a very supportive supervisor, and also Ralph, who hosts our group at his chair and also supports all our projects a lot. Then, of course, my group and Tina and her team for doing the MS analysis and creating these interesting results for us. Yeah, and if you have any questions, feel free to ask.
Thank you. Thank you very much to you for to give me the chance to present some of my work. We are really looking forward to get some feedback. This is the first time we are showing this work to anybody. So if you have any questions or any recommendations, we are very welcome. And so far, we are using, but to make it in order. First, I want to make sure we have some common ground. So very extremely short introduction to mass spectrometry. Then I'm gonna get into the topic of this software that we developed, OrbiFragsNets, with the fragmentation tables. It's kind of an internal object on the data processing. And then going directly into OrbiFragsNets. Hopefully it's very easy to use. Then we developed the framework with the Orbitrap high-resolution mass spectrometer for those who want to see what's doing an Orbitrap. Here it is. It's taking a molecule, a chemical, and it's extracting some information. That's the MS2 spectrum. So you can think about it as a process of breaking apart. So we have our molecule, a mixture of different pieces. Then we break it apart and we see the small pieces. You can think about it as a fingerprint as well. Is an MS2 spectrum is unique. So you can, for example, do your analysis with your unknown chemical, get your spectra and match it with databases and confirm you have the same chemical. Then you have some information, the intensity, which would be related to the stability of those fragments, and the mass to charge ratio, the size of those fragments. Then we can improve the quality of that information by annotating. We can give a suggestion of the molecular formula of that chemical. So for example, here, we did the process of annotation. Well, yeah. In the spectrum you see on the left, and we did the process of annotation, and we can say that those fragments maybe, thank you, maybe would correspond with that molecule. Then you might wonder, how do I get from the spectrum on the left to the spectrum on the right? Because we are changing the information from numbers to a chemical composition. So we can move the other way around pretty easy. We take the molecular weight of each element, we add it up, and then we get this value. But we can we do it in the opposite way? There are some work already done, and this is very popular. Maybe some of you have some experience with Sirius 4, the last version. And they are based in Germany. Actually, they do this process of automatic annotation using a machine learning approach. So maybe it's not so good for unknown chemicals. Maybe it is. Well, they're working on that. But also, I would say they have the limitation that this is an external platform. It's an external software. So it's difficult to integrate when you want to analyze large volumes of data. We have a spectrum, just coming back. I'm showing the same data as before, but as a table. So we have the mass to charge ratio, the size of the fragments, and the relative intensity. Then we annotate the spectrum, and we have some chemical information. However, I think that some more useful information we can include in this table. For example, what about those values? We always get those values as fixed values. So a mass to charge ratio as a fixed value. Probably all of you know that everything have an uncertainty, and it's very interesting that it's not very discussed in the mass spectrometry literature. So we think we need to include a confidence interval to do some statistical treatment. Then, of course, you would include the number of samples and the contrast with the model. So we make a prediction of the molecular formula. We can calculate the predicted value. We also have a measured value, and we can evaluate. And just to point out, those data points are not referring to remeasure the same sample over and over again. Actually, we propose to extract all of that information only from one sample. because we usually think that a mass to charge is a fixed value, but actually there's a distribution. We can use some descriptors on these distributions. So the average value, which would be the value that always everybody reports, and they get it from a centroiding process. But you can also describe the distribution with the standard deviation. You can even do some statistics. You can estimate the confidence interval and think the annotation process as a statistical test. So I have the confidence interval for this mass to charge ratio. That means that the real value should come from the molecular formula whose value is inside this interval. So I can do the same process for all of the signals in my spectrum. And then I would have a table with a list of candidates for annotation on each fragment. How do I define that is this one and not this one? Then this is the key aspect of this data processing approach. And we think that the fragments are not independent. They all are coming from the same molecule. Then as they both are ions, they should be related by the chemistry. Then we think, okay, if we can correlate two fragments and two suggestions for fragments. So here, every colorful ball is representing a suggestion for an annotation of a fragment. Then if I can express the relationship between two of them and a neutral molecule leaving the reaction, I would say that they are correlated. They are from the same spectra. So they are from the same molecule. We call it chemical consistency. They are validating each other somehow. Then we can suggest a series of them. I can make a list, but that's not the only option. I can make several possible combinations of all the possible annotations. So for this example, I'm including here, I could annotate 4 fragments, and in each of them, I have different possibilities. So 4 times, 3 times, 2 times, that's 24. That means I have 24 possibilities to describe this spectra. Which one is the real one? We use the chemical consistency as a criteria for annotation. And might sound a bit complicated. Hopefully, if you are curious, you can check the paper. There we are describing, well, this is a preprint. There we are describing the confidence interval with some interesting references and the molecular, the network that we built with those fragments. We also have everything already available in the GitHub repository, and you can check the link, download, try it. That would be very valuable. And even more interesting, it's super easy to use. So you just need to format your data as MZ-ML extension, or usually from OrbiTrap, you get them as a raw file, but you can use MZ-convert and move to Python. Then here you charge your data, and you need to know the molecular mass for your target. But if you scan all of your features in your data set, you can get an annotation for all of your possible chemicals. Then you annotate it, just everything that I described, the steps, it's happening inside this function. And then you get a table with the results I just described. Then thank you very much for your attention. Looking forward to hear any comments or questions from your side.
Thank you for your...
We can talk. Now it's quite, relatively easy. Just tell you how to do the biomarker research based on the proteomic. So I will talk to you about the head neck. What is head neck? Maybe one of us will get in the future, maybe not. And what is extracellular vesicle? All the time called the exosome, but nowadays talking about the extracellular vesicle. So you will understand more why the extracellular vesicle is important in the circulation of the blood. Okay, yeah. So head and neck cancer is the sixth of the common cancer in the worldwide. So in the 2018 will be 800,000 will annually get the head and neck detected by the physician, and predict that 2030 will over 1 million people will get the head and neck cancer. And the 90% of the head and neck cancer is related to the head and neck squamous cell carcinoma. So it's detected by the, it's also have the other type of the cancer like the adenoid cell carcinoma or something like that. But in the head and neck cancer, majority is the squamous cell carcinoma. Yeah, and then the 50% of the people 5-year survival rate is very low, and also another 50% of the patient have the really high chance to get the recurrence. That is mean if you remove that by the surgery or you do some chemical therapy, but later on you will get the cancer again. Yeah, and so reliable biomarker is urgent need for the physician to understand for the diagnosis for the therapy and the therapy topic, which as you can might hear before, like the PD-L1 therapy, antibody therapy is more famous recent day. Yeah, but it's not good enough. Yeah, so why the tumor is so complicated to kind of cure? Because tumor involvement is relatively so complicated. It's called a tumor microenvironment. What is inside the tumor microenvironment? So we knew that we have the tumor inside, but of course we also have some immune cell. So it's called a tumor infection, tumor related to the tumor related immune cell, such as the macrophage, dendritic cell, and T cell. So a lot of people studying the immunology, they focus on why those immune cells are helping the tumor growth. Okay, but we are in a different way. We are investigating what is secreted outside the tumor. Okay, so because the tumor cell and also the immune cell will secrete the small molecule, like a cytokine, like a protein, like a secretome, and also there's one kind of vesicle will secrete. Like it's called the extracellular vesicle, the older time is called the exosome, and also the microvesicle. Exosome is around, I will show you later on what is the size of the exosome. And in general, like those cells, majority of the cell, 100% of the cell, even the plant, even the bacteria, are all secrete small vesicle. Okay, so there's now it's getting more hot topic in recently. So you can detect from the biofluid and detect from isolate from the biofluid about the exosome. Yeah, and the cell, how the cell secrete the exosome. Yeah, the cell will internalize forming the early endosome and then to fuse inside to forming the intraluminal vesicle, and those multiple vesicle body will fuse with the cell membrane and then release the exosome. And what is inside of the exosome? They contain the protein, nucleic acid, and metabolites. Yeah, so those things we can do the NGS to sequence the microRNA, to sequence the messenger RNA. We also can send to the TINA to make their work more harder to do the proteomic. Yeah, that is what I'm doing. I send a lot of sample to them. Yeah, and then what is important marker for the proteome on the exosome? It's called a tetracycline family. So if you want to read the literature future, so there's 3 markers very important. It's called CD9, CD63, and CD81. You will often hear from the human extracellular vesicle. And inside is like Alex, this is helping the EV secretion. It's called the EV biogenesis. Yeah, also the some potential surface marker on the surface, such as the PD-L1, APCAM, or the pan-CK, that is kind of the tumor-related protein. Yeah, but in exosome field, actually we're facing a bit challenge, because if we look at the size of the exosome size is around 30 to 200 nanometers. So actually the SARS-CoV-2 is also the similar size. you can think about that if we infect the virus in the like influenza or rhinovirus or COVID. So actually in our circulation body we have the virus. We also have the vesicle. Those vesicles are share the similar size. Once we want to isolate the EV, so the golden standard method is ultracentrifugation. But ultracentrifugation is not only isolate the acetone, but also isolate the lipoprotein and also the virus as well. So oftentimes we actually got a lot of contaminant. So we need a kind of reliable isolation method. Now have the like a size fusion chromatography or some antibody-based isolation. And what we are using now, we are using our in-house developed. We recognize the glycan on the EV surface because we knew that a lot of the surface protein are glycoprotein. So we can write they have some specific pattern compared to the lipoprotein and secreted protein. So we can use in the specific pattern and we coating it's called a glycine coating on the magnetic beads, and we just incubate with the plasma, human plasma, or some urine, saliva, it's all fine. So then we got the EV complex, and we license it with the RIPA buffer and amina, and we send to the core facility, and using, then we do the data analysis. And after we got the candidate, we have to validate the data if those proteins are really expression on the EV surface or intracellular. And later we're using the bead-based flow cytometry to further demonstrate that, yeah, we actually identify those target proteins. Yeah, so here is just let you know if we got the EV isolation, what it looks like in the cryo-EM. This is a cryo-EM. So actually we have some small vesicle, the size is around 50 to 200 nanometer, and we have to can see the double liquefied layer on the EV surface. If we send the immunocoat scanning, actually we can see nicely they bound to the surface because the pan-EV, as I told you before, 9, CD63, and 81, they can recognize the surface of the EV, so we can get very nice image from the immunoquality. And the nanocheck analysis show us that the size of what is our isolation is between 30 to 200. That is exactly the size what the literature report. So that is mean we can get quite pure exosome from our plasma. And later on there's two kind of application I want to show you. One is we already did some small paper publication. It's a pilot study. So we compare two group of the patient. One is a relapse patient. Another one is a remission. Remission is more or less, oh, this patient is stay healthy, no tumor can form, something like that. But relapse is mean this patient at certain time point, the tumor is coming back again after the surgery. So that is why we can have to investigate why some patients still keep healthy after the therapy, but why some patient is still tumor will still coming back. Yeah. And the second one is what is our ongoing work. So another one is okay. Another idea is okay during the decision because my another doctor father is in the University of Boston. So we're just talking about oh okay maybe we can take the blood during the surgery because we can check if the marker something changed before and after surgery. Yeah, then we can think about that. Okay, those markers are reduced significantly. Then those markers could be a good prognosis. We can also fit into the first study as well. So this marker could be a good prognosis marker in the future. If the tumor, if the patient have the relapse, maybe this marker will coming back again. So that is why these two projects I would like to share with you. The first one is collective. Okay, so we send around 9 sample, 9 donor. It's not that much, so we call the pilot study. So 4 from the relapse patient, 5 from the remission patient. And then in general, we can talk here around 400 to 500 EV protein. Yeah, so compared to the cellular protein, this protein rate is relatively low. Yeah, and we can also target some significant protein. And I just point out that in relapse protein, we can target some complement like C1qa, QB, QC. And also another group of the protein are collected. Collecting is also one type of the lectin. And what then we do the gene ontology, protein ontology pathway analysis. So we can target some like a collagen and also some extracellular region. So for example, the collagen is important because as we know that in the tumor microenvironment, collagen will also help them to kind of grow very well. Yeah. But further on, we have to demonstrate that the collectin 10 is really increased in the relapse patient. So we check back the raw data from the mass spectrum. So intensity is really significantly reduced. And further on, we introduce the bit-based flow cytometry because the collectin 10 is an intracellular protein. So we do the intracellular staining in the EV. So we can see the intracellular compared to the surface staining, they are a bit increased. Then the further on, we want to test in the different patient that, oh, okay, this collectin 10 is really high in the relapsed patient. Yeah, so in here we combine our old standing marker. We have the several old targets, a well-known common marker in the head and neck. One is EpCAM, another one is a PD-L1, and another one is a pan-CK. I'm not showing here. So the result is quite interesting. Collectin 10 is with the tiny double positive, we can see the significant reduce from the remission patient compared to the relapse. And the pan-EV positive, CD45 negative, and collectin 10 positive. This is kind of why we stand the CD45 here. CD45 is a lymphocyte marker. So if we combine because the tumor cell is a CD45 negative, lymphocyte immune cell are all CD45 positive. That is why we can kind of confirm that is our EV is really isolated from the tumor. It's not from the immune cell. So this is really important. That is, we show the people that this tumor secrete exosome is really in the relapse patient is really expression high collectin 10 compared to the remission patient. Okay, yeah, this is, and then yeah, so this only tell us, yeah, collectin 10 is really important. Might be important because it's not that many donors. It might be important in the relapse patient. However, we have to link to the maybe, yeah, the clinical database is if the collectin 10 is really important in the like a patient survival rate or not. So we do some research. So I think you are very familiar with this database. So we searching that is if the collectin 10 higher and lower have some differences or not. Yeah. Then we kind of luckily we found that is in the female patient, if the female expression collectin 10 high expression, then the 5-year survival rate is significantly lower compared to the low expression female patient. That is maybe, but it's just the kind of the preliminary trial. We have to collect more a large cohort for those study. So it can suggest that is collectin 10 maybe important, especially in the female patient, the female relapse patients. Yeah. And next study I want to introduce you is what we are is ongoing work is before, as I mentioned before surgery and after surgery. We collect the peripheral blood and also the local blood. And peripheral blood is mean if you go to the doctor, they took from the venous, and then we got the blood. That is called the peripheral blood. It's circulation. And local blood is mean local venous plasma is very when the physician do the operation, and then they have the one major venous blood, so they collect the blood from there. Yeah, why we do that? Because of we think about that is maybe the biomarkers are diluted in the circulation because we still have a lot of cells secrete the EV at the moment at the same time. So, but from our result, we found quite interesting. Only we can find some interesting stuff from the peripheral blood. PP1 is mean before the operation. PP2 is mean 1 hour we collect the blood in peripheral and the local. And then in general, we can target around 360 to 400 protein in 4 group. And some unique protein we can also identify. And then we perform I using the pursuits to do the data analysis. So I filter, I want to kind of get the more specific protein. So I did a very strict filter. So I filter out everything, every sample must be expression those protein. So it's only around 200 protein are in the analysis. So we can target, we still can target 6 of protein and 1 protein particular is very interesting for us. It's called the integrin beta. Okay. And we check again the original file from the mass spectrum, the intensity. Integrin beta 3 is significantly reduced after 1 hour operation. And we also perform the B-space flow cytometry. So again, CD45 negative and EpCAM positive. That is mean really tumor secreted isotope. And plus our target protein is integrin beta 3. So integrin beta 3 sometimes also called the CD61. So we can see that from the ball donor is a significant reduce. And what is the pathway behind the integrin beta 3? So we can find some kind of ECM related and the cell migration related because of the integrin is on the surface and then help the cell to do the migration. Yeah. And yeah, so again, so we check the database if the integrin beta 3 is really important in the large cohort. So those are the RNA-seq data. So we also saw that is when the patient high expression integrin beta 3 compared to the low expression, the 5-year survival rate is also significantly different, but not really high compared to the collective. The female collecting result is around 10% less, but the p-value is still significant. But we still collect more patients. We want to collect at least 30 or 40 patients in general. Yeah. So those are what we are doing now. And thank you for your attention. And yeah, thank you for Tina's and the chat in the Zoom. So to help me to do the proteomic and the data analysis. Yeah. Yeah, thanks.